Hey science fans, I'm going to make this little video about how to use Project Discovery. So I've made a blank character for you and I'll go through some of the tasks and talk about how I think they might be classified and what sort of logic we use here at the HPA. So first just open Project Discovery up. Yes, start tutorial. And I'm going to zoom through these because I've already read all of them. You can read them at your leisure. They're going to explain what the different categories are. Here you can change colors and submit things, your rank, accuracy, and how many you've submitted. You can also go back to it. Yep. So this is the basic interface. I can zoom in on things, click to lock, and I can see where I am here, and I can toggle channels on or off. So in this particular sample, you can see pretty clearly that if I turn off the microtubules, the red, there's not much green outside of the blue. Whereas if I turn off the blue, then we see a lot more signal. And if I turn off the blue and the red, we can see signal where the blue was. So it's somewhere in the nucleus. Next we have to check whether it's in the nucleoplasm or whether it's in the nucleus as a whole. And for that we want to look and see if we can identify where the nucleoli are. Nucleoli are these dark areas inside the nucleus. So if I turn off the blue, can I see those? And not very clearly. So I'm going to choose nucleus. It's a little bit slow here on my laptop, but I couldn't use my desktop in the lab because it couldn't disturb everyone else trying to do some science. Alright, so here you can see a little explanation. I got this one correct. And here you can move on to the next sample. So once again, I'll toggle on and off some channels. This is clearly in the nucleoplasm, but you'll notice that it's not an option here. So if we look at some of these other options, it's not in the nucleus because it has clear holes in where the nucleoli are. And turn on the blue channel and turn off the green, you can see here the nucleoli. And you right away see the holes where they are in the green. But it's not nucleoplasm either, so what could it be? Well, this is actually nuclear speckles, so if you hover over these you'll see some examples here. And it looks pretty similar. It's sort of this variegated pattern of green inside the blue. So I need to select that, and I can submit. Okay, this one looks as though some sort of cytoplasm. It's punctate, so you might want to pick vesicles, but really here there's far too many dots for it to actually be vesicles. This is just a punctate pattern in the nucleus, or sorry, in the cytoplasm. I'll choose cytoplasm, and I don't see any of these other things in here, ER or bridge. And for these initial samples, remember that you can always check out what it says here, granular cytoplasm. This is not available for the real-world samples since some of them we don't know the answers to, and a lot of times we can't really tell you why you got it right or wrong, or even if you got it right or wrong right away. So this one is a typical MTOC, and the reason that it's MTOC is because you can see here where the red are kind of all coming together. So if I turn off the green, I'll turn off the green for a second. You can see the red all come together into this one area, and we have sort of a spattering of green there. Now we can look at a couple other cells to confirm that. So you see here also where we have red microtubules coming together and green on top. But it's not centrosome because typically centrosome will be these two dots right next to each other, as you can see in this image or this image. Whereas mTOC will be in a similar location, but will be more of a spatter. So I'll select the mTOC. Sorry, mTOC means Microtubule Organizing Center. That's our in-house abbreviation, since that's very long to say. Okay. This one's a very clear vesicle pattern. Just get one cell in focus here so you can see. It looks pretty much spot on to what a vesicle should look like. Sometimes there's fewer and sometimes there's more, but it shouldn't be totally filling the cell like that granular cytoplasm we saw earlier. Alright, this is a typical plasma membrane, really nice. This is because you can see the fingers coming out here where the membrane is actually reaching out and holding onto the substrate. Okay. This looks like a mitochondria. It's sort of spaghetti-like. So unlike vesicles here, the pattern is not clearly little dots. It's more strings in the cytoplasm. And so that's indicative of mitochondria, but you can see some of the cells, like this one here, don't have any mitochondrial staining at all. They have mitochondria still, all cells do, but this is a cell-to-cell variation. So make sure to check that off. Now you may think that I'm a lot better at this than you are, but don't get discouraged. I do this all the time, and also I helped make these samples, so of course I already know the answers to a lot of them. So in this one we see again this clear fingering out on the exterior of the cell. That's plasma membrane. But we also see some nucleus and maybe even nucleoplasm here. So as you can see, maybe this is a nucleoli. I can clearly see the outline of the nucleus without the blue channel on, meaning that I should probably mark something about the nucleus. So I'm going to choose nucleoplasm. I might be wrong. Oh, got it. So in this one, we see really nice nucleoli here. And you don't get the really brilliant holes in the nucleus for this particular cell, but these should be nucleoli because they're big and there's not that many of them. And we also see these hair-like structures here. And they don't overlap with the red marker, the microtubules. So these are intermediate filaments. One cool thing here is that you can tell that this cell has nucleoli, but no staining on the intermediate filaments. Whereas some cells have a lot of staining. So this is again cell-to-cell variation. Cell-to-cell variation might be indicative of different stages of the cell cycle, or possibly different conditions in prep, although that's less likely since these are clonal populations of cells. Oh, this looks like a pretty ugly staining. Let's look at green. So here you can see that if I toggle on and off the blue channel, I have no idea where the nucleuses are, the nuclei are. So I couldn't circle the outline of them. So in this case, it shouldn't be both nucleus and cytoplasm, but rather just unidentifiable. This is a trash signal, most likely due to just some background of the antibody getting stuck inside the cell. Alright, I hit rank 2 and move on to the training phase. And these we also know a correct answer, so it's still a little bit easier. But now you're able to select from the full set of categories, which makes it a lot harder. So this could be bodies, or could be nucleoli, but to me it's very clearly nucleoli because of the size and the number here inside the nucleus. Let's just check the blue channel, and you can see a hole here right where we have a bright green stain. So nucleoli it is. And just zoom out, make sure we don't have any other staining. I think we're good. I'll get some points. These are pretty cells, but the staining doesn't look that pretty. It's clearly not very much in the nucleus. So if we look at the nucleus, we don't see a lot of green overlapping it. But it is definitely in the cytoplasm. So just going to mark cytoplasm for that one. That's a small bug in the interface there because of my laptop not loading things very quickly. So this one is very clearly similar to this pattern that we have here, which is mitochondria. So we'll choose that one. That's not right, yeah. And again, I'll just toggle the green. This is really useful to do because you want to make sure there's nothing inside those nuclei, which we see really nicely. Good signal to noise here. Lots of signal in the mitochondria, not much elsewhere. Kind of late. Alright, oh, this is one of my favorite patterns. These are focal adhesions. You can see they look sort of like little, I like to think little paintbrush strokes, little splotches, usually on the edge of the cell. These are holding the cell down. So the cells are grown on a plate, and they grab onto it to hold themselves in place. But you can tell the difference between cell junctions and the focal adhesions because the cell junctions only occur between cells. So you shouldn't see cell junctions occurring other places, only between the cells. Whereas focal adhesions can occur kind of out in the middle of space. And that's sort of what we see here. I mean, this cell has focal adhesions here in the middle of nowhere, not touching any other cells. Sometimes you have focal adhesions also underneath the cell, and that's what you're seeing there. So it's focal adhesions and not much else. Alright, this looks like a pretty nice ER sample to me, where we have this sort of spiderwebbing pattern. You can usually really clearly see the nuclear outline in these ER samples too, which is really nice. Now we look here with the microtubules on. It usually fills a large part of the cell as well. Oh. Okay, so rushed through that one a little bit. Should have looked closer, but yes, you can clearly see nucleoplasm in here. And of course, do the check where you see the nucleoli and the hole in the green staining. I'm trying to get to some of these uncharacterized samples for you, which are a lot more interesting. Okay, these should be vesicles. Let's not make the same mistake I did last time. Rushing through as I go really fast. Okay, I got it that time. This is a Golgi pattern. You can tell because it's hugging right next to the nucleus here, and typically on only one side of the nucleus, not surrounding. So the Golgi pattern is here. Yeah. I can look at a couple other examples and confirm that it looks very similar to what we're seeing now. And just make sure I'm missing too much. I don't think so. Now you may say there's a little bit of background here in the nucleus, these spots, but it's so much dimmer than our actual signal, and it's not very many, not very bright, and so this is likely just some antibody getting stuck in the nucleus. Usually we don't annotate if it's much, much dimmer than the actual signal. Alright, this is an interesting one. This might be my first foreign one. Oh, it looks like some sort of vesicle, or maybe more accurately, a type of cytoplasmic body. These are a bit too big to be vesicles, and there should be more of them if they were actually vesicles. But we'll choose vesicles here since we don't have a cytoplasmic body. Just double check that it's not centrosomes or microtubule organizing centers. But the dots look clearly not aligned with the center of the microtubules. So I think I'm happy saying that it's a cytoplasmic body or vesicle. Okay, it was an unknown one. And it looks like most of you are agreeing with me, so that's pretty great. A lot of people are confused on this one. Like I said, it's kind of not fitting a lot of other ones. Some people said agrosome. Agrosomes are typically a little bit bigger than this, and they actually usually are one per cell and not multiple. So that's why I didn't pick agrosomes here. Alright, so we're into the unknown, the great unknown. This is pretty clearly nucleoplasm. Just want to double check because, nuclear speckle is also a possibility here. It's a little bit variegated. I should zoom in to see. But it's not looking like a leopard speckles, really. I think it's a little bit too smooth here to be considered speckle. So I would choose nucleoplasm. And looks like everyone's agreeing with me on that one. Okay, this is kind of an ugly one. Look at this little cell here. Something's happening. This is either a cell dividing or maybe exploding. Sometimes cells get damaged in preparation. And so typically, because we see that, even though there's a bright signal there, we're not going to care so much about analyzing that signal, because that cell looks so abnormal. But this looks like a vesicle staining to me. There are a lot of dots here, but they're pretty well spaced. It's not really filling the cytoplasm, so I wouldn't annotate cytoplasm. I'd pick vesicles for this one. Okay, some disagreement here. Some people think there's too many dots to be vesicles, and some of you also chose the nucleus. So let me just look. I mean, here's the thing. Sorry. Here's the thing about choosing nucleus. Typically, you should be able to see exactly where the nuclei are if you're going to pick it. And in this case, I just don't really think that you can tell me that there's a strong signal in the nucleus. So for me, it's not nucleus, but I can understand why some people thought that it would be. It's good that other people are agreeing with me, so it seems like you guys are actually getting this. So it makes me really happy. Oh, this is a good one. Okay, this is centrosomes. And you can tell right away. You see these two dots right next to each other, and they're right where all of these red lines are coming together, the microtubules are coming together. This isn't MTOC like we had earlier, which is a splotch. It's the centrosomes indicated by the two dots right next to each other. So that's a cool one to get. They're not so common, not too many proteins in the centrosome. And double check the green channel. Okay, there's some other stuff going on in the cell, but it's not a lot. It's most likely just background. I'm not going to be surprised if some people choose to say that there's cytoplasm or something, but again, I can't tell where the nucleus is in this view, so I'm not going to annotate both cytoplasm and nucleus, and I shouldn't annotate cytoplasm if there's no hole where the nucleus is, and I shouldn't annotate nucleus if I can't see the difference between it and the cytoplasm. So to me, this is just some extra background staining that the antibody got stuck a little bit, but some very, very nice centrosomes there. And it looks like some of you are thinking about what I was saying here. cytoplasm. And some people thought nuclear bodies. Now, that's tricky because it looks like these are in the nucleus, but if you turn on the red, you can actually see. So remember, we're taking slices through the cell, and sometimes we're a little bit above or below the actual nucleus. So interestingly, it looks like I might have been the only person to pick centrosomes. A lot of people thought vesicles. Again, I think just because they're so much dimmer than the centrosomes, I wouldn't really pick it, but you can. And we do encourage you to select things that are even a little bit dimmer. We'd rather you over-annotate than under-annotate. Interestingly, a lot of people thought this is unidentifiable. Just keep a lookout for these small features. Some of these features are really small, like the centrosome and also the cytokinetic bridge is a really fine feature that there might only be one of in the image. So make sure you look at the whole image before making your pick. Alright, this is a clear cell-to-cell variation. You can see some cells are expressing this protein a lot, and some cells are not expressing it at all. As to the location of it, it's definitely not in the nucleus. So if we toggle on and off the channels, you can tell it's not in the nucleus for sure. It looks to be pretty clearly, smoothly in the cytoplasm to me. It's kind of, you can see it's a little bit ugly here that the red is not uniform throughout here. That's because these cells are kind of growing in a clump, and you can't get the staining into the cell group as well as if the cells are separate. There's just less surface area, and so less ways for it to go in there. And okay, people are really agreeing with me here. It looks like you guys are getting the cell-to-cell variation too, which is really good because we haven't been annotating that here at the HPA very much, so that's a big contribution that you guys will be giving us, along with the subnuclear patterns. All of these up here we haven't done before for many of them. Alright, so my first thought on this one was not identifiable based on the red, green, blue. Just didn't look like much. When I put on the green only, I see something interesting down here in this cell. Something weird's going on here. I need to take a closer look. Oh, it looks like right on the edge of the nucleus there. Just a little extra. But it's corresponding with this ball in the nucleus, which is probably just sort of a concentration of this background on a crappy staining. So let's zoom out again. Look at the green only. I thought I also saw something up here. Maybe mitochondria, if it was more spaghetti-like. Now, for us, we actually would look at other cell lines before putting it on the atlas, and we would be able to tell maybe this particular cell line doesn't express this protein that well, or maybe there's some literature to tell us that it might be mitochondria, so this borderline case we would get some more information on. But in this case, I'd rather take a guess at a location than guess not identifiable. I mean, not identifiable, it's kind of a cop-out for not being able to tell something, and I always try and annotate something rather than nothing. So I'm gonna take a guess here and go with mito, even though it's really borderline. Very borderline. Could easily annotate this not identifiable. person that picked that. Some people said cytoplasm, some people said nucleus, but a lot of people thought not identifiable, and that's fine. Like I said, I'm pretty confident that that one will go to not identifiable. Alright, this one's got some really bright signal in one cell over here. What's going on here? Some bright punctate nucleus. Yeah. So, it's definitely not nuclear bodies few. It could be nuclear bodies many here. Typically we see not a huge amount. This is more just a punctate nucleus or nucleoplasm actually. If we turn on the blue channel, you can kind of see, I'll get a better example here, some holes in the blue, but they don't, the holes in the blue don't appear to be correlating with where we have some missing signal in the green. So I'm just going to say nucleus for this one. And maybe some cell-to-cell variation just because of that one cell. I'm the only one that cared about that one cell. Poor little guy. Okay, this will be the last one that I'll do for you guys. I'm going to get back to answering some questions over on Reddit. This one, it's pretty hard. Looks like it's definitely in the nucleus, in the nucleoplasm, but possibly some speckling. Let's turn blue back on. Like this cell up here. That could be a nuclear speckle pattern easily. We look at our other examples. You can see it's pretty similar. I personally always have a hard time with distinguishing the nuclear speckle because it can be so similar to the nucleoplasm. There's also definitely cytoplasm in there, so let's click that before I forget to do it. If I turn this off, I wish I could click that mini map and go to where I want to go. This is interesting. You can actually see a nice line around the nucleus. That would be nuclear membrane. So actually I'm going to choose nuclear membrane because I thought I saw that in a couple other cells. Like this one here. Really nice line. Where it's dimmer on the inside and the outside. So really bright line around the nucleus, that's going to be nuclear membrane. And for me, it's just not variegated enough to be speckles. So I'm going to choose nucleoplasm after all. And I think that's it. Let me just make sure that I don't think it's plasma membrane. This cell here made me think maybe it could be. You can see it's got some of these fingerings, but then I looked down at this cell and I thought maybe it wasn't, because this extends past. So, it looks like I'm getting done with this at the right time. So there's going to be some temporary downtime. Let me just make my selection here. Okay, I think I'm happy enough with that. Let's see what you guys thought. Okay, most of you agreed with me here on the nucleoplasm and on the cytoplasm, but none of the other people who had this image saw the nuclear membrane, so just keep an eye out for that, where you have an area that's brighter along the edge of the nucleus than it is both outside and inside the nucleus. It's pretty tricky in this one. Obviously, this example here that we chose for you is going to be a much clearer one. Sometimes you see these squiggles in the middle where you have some membrane folding. And don't be fooled, those aren't bodies, those are actually just pieces of membrane that are folded up. The nucleus isn't a flat organelle. It has a lot of wrinkles and stuff in it. So yeah, just keep an eye out for that. And that's been sort of my little explanation walking through some of these in Project Discovery. You can see that I'm not always perfect at this either, but hopefully together we can get some good results, and it looks like you guys are already well on your way. Thanks.